Hi friends, welcome to X channel. In this video, we are going to see 10th Standard Science Unit 18 Genetics Part-III Additional Questions. 4th Roman letter short answer question. Define a gene. Genes are segments of DNA which are responsible for the inheritance of a particular phenotypic character. Inheritance of a particular phenotypic character. State the law of dominance. Law of dominance. When two homozygous individuals of one or more sets of contrasting characters are crossed in the F1 hybrid, the character appear are known as dominant and those don't appear are recessive. This is law of dominance. State the law of independent assortment. In the inheritance study of two or more pairs of characters simultaneously, the genes of one pair assort independently of the other pair. Why do we call law of segregation as law of purity of gametes? Gametes are pure for a character because they are always haploid. During gamete formation, alleles of a pair segregate such that a gamete receives only one allele and so it is pure for a character. Differentiate between dominant and recessive conditions. Dominant condition. When an allele expresses itself in the presence of its recessive allele, it is called dominant. Please like, share and subscribe Leela Kids channel. இந்த வீடியோவை உங்களுக்கு பிடிச்சிருக்குன்னா லைக் பண்ணுங்க, ஷேர் பண்ணுங்க. again and again போட்டு பாருங்க. எக்ஸாம்ல உங்களுக்கு ஃப்ளோ ஈஸியாக இருக்கும். ஆல் தி பெஸ்ட் ஃபார் யுவர் பப்ளிக் எக்ஸாமினேஷன். அண்ட் தென் இது மாதிரி ஆன வீடியோஸ் உங்களுக்கு ஷார்ட் வீடியோஸும் கொடுத்துட்ருக்கேன். அதையும் பாருங்கள். again and again போட்டு பார்க்கறதுனால ஆன்சர்ஸ் மெமரைஸ் ஆகும். Differentiate between dominant and recessive condition. Dominant condition, when an allele expresses itself in the presence of its recessive allele, it is called dominant. Recessive condition, it can only express in the absence of the dominant allele and remain masked in the presence. Name the enzyme involved in DNA replication. DNA polymerase, DNA ligase, helicase and topoisomerase. Draw the four types of chromosomes based on the position of centromere and add a sentence and describe each. So this is the diagram 1, 2, 3 and 4. Refer text for this diagram. Acrocentric rod-shaped centromere at one end, a short and long arm. Submetacentric centromere near the center, two unequal arms. Metacentric centromere on the center of the chromosome form two equal arms and V-shaped. Telocentric centromere on the proximal end rod-shaped. Differentiate type questions. Distinguish between karyotype and ideogram. Karyotype, the number, size, shape of chromosomes in the cell nucleus is known as karyotype. Ideogram, when karyotype of a species represented in a diagram, it is known as ideogram. Distinguish between Down's syndrome and sickle cell anemia. Down's syndrome, it is a chromosomal mutation. Sickle cell anemia, it is a gene mutation. Here there is an extra chromosome. Changes in nucleotide sequence of a gene or point of a chromosome. Mental retardation, delayed development, vision and hearing, disability, behavioral problems, etc. Changes in the gene affects the proteins produced leading to sickle-shaped RBC that carry hemoglobin. Long answer question. Explain different types of mutation. Phenotypic changes observed first by Hugo de Vries in Oinothera lamarckiana. Term mutation was coined by him. Definition: sudden change in the genetic material of an organism. Two types: chromosomal mutation, gene mutation. Chromosomal mutation, sudden change in structure or number of chromosomes. Change in structure of chromosome and changes in number of chromosome. Change in structure of chromosome occur due to error in cell division, and change in number of chromosome involve addition or deletion in number of chromosomes known as ploidy. Change in number and arrangement of genes resulting in deletion, duplication, inversion, translocation. It is of two types, euploidy and aneuploid. Euploidy and aneuploid. Euploidy more than usual number to n, aneuploid loss or gain of one or more chromosome in a diploid set. If three haploid sets, triploidy 3n, triploidies are usually sterile. 2n plus 1 trisomy, 2n minus 1 monosomy, 2n2 nullisomy. Example, Down's syndrome. It is aneuploid, 21st trisomy. Symptoms, mental retardation, delayed development, behavioral problems, weak muscle tone. Gene mutation or point mutation, change occurring in nucleotide sequence of a gene. It involves substitution, deletion, insertion or inversion of a single or more than one nitrogenous base. Sickle cell anemia. Alteration in a single gene brings a change in the structure and shape of protein or BC. Explain process of DNA replication. Refer text for diagram. DNA molecule produces exact copies of it during replication. The nucleotide of each strand act as template to form a new strand. Origin of replication and unwinding. The specific sites where replication begins is known as site of origin of replication. Heliocase. The two strands separate at this point, forming replication fork. It separates the two strands to polymerase, separates the twist above the replication formed during unwinding. RNA primer. Short RNA nucleotide initiates replication. DNA polymerase help in the addition of nucleotides and a new strand complementary to the template is formed. In the leading strand, synthesis is continuous. In the lagging strand, short segments called as Okazaki fragments are formed, later joined together by DNA legacy. At terminus, the replication fork of two sides meet and replication stops. So, இந்த வீடியோ பிடிச்சிருக்குன்னா சேனலை லைக் பண்ணுங்கள், ஷேர் பண்ணுங்கள், சப்ஸ்கிரைப் பண்ணுங்கள். இது வரைக்கும் சப்ஸ்கிரைப் பண்ணவங்க எல்லாருக்கும் தேங்க்ஸ். இந்த வீடியோ again and again போட்டு பார்க்குறதுல ஆன்சர்ஸ் மெமரைஸ் ஆகும். We can meet in the next video. All the best for your examination. Bye bye.